I am here at ASH to present data from our first-in-human phase 1 study of the REGN1979 bispecific antibody. This is an antibody that binds to both CD20, a target on B-cell lymphoma cells, and to CD3 on T cells, and it works by activating the T cell, redirecting the T cell to the CD20 expressing B cell, and killing the lymphoma cell with specificity to CD20, independent of the T cell receptor's own specificity. We have completed the dose escalation part of our study, and so we're reporting our final results on the dose escalation. The study does continue with expansion cohorts, and those are being enrolled too. This drug is given in a step-up dosing manner, where we begin with a low dose in the first week and step up gradually over the next 3 weeks to the final dose in the 4th week. And using the step-up dosing manner, we've been able to control the toxicities such that we have not experienced any dose-limiting toxicity in our B-cell lymphoma cohorts, and we've taken the study to the maximum dose that was written into the protocol without any DLTs. The main toxicities that we see are those that would be expected for this type of bispecific antibody. Those include infusion reactions and cytokine release syndrome. We see those typically in the first few weeks of treatment, and for the most part they are grade 1 or grade 2 toxicities, very few grade 3 toxicities. By the 4th or 5th week of treatment, we see only a smattering of grade 1 or 2 toxicities, so it's very early on. We are also on the lookout for neurotoxicity, as that's been reported in other medications using a similar T-cell activating mechanism. And what we see is primarily grade 1 and 2 neurotoxicities, typically things like headaches, insomnia. We've seen a few grade 3 toxicities, no grade 4 toxicities, no seizures, and so we feel from a neurologic point of view it's quite well tolerated. This is a phase 1 dose escalation study that's basically looking to define a safety profile and looking for a dose, but we do capture response data as a secondary endpoint, and we have been pleased to see activity in these very heavily pretreated patients. The median number of prior therapies for the lymphoma patients on the study are 3, at least 3 prior lines of therapy, and or I shouldn't say at least, a median of 3 prior lines of therapy. When we look at the patient population, about 2/3 of our patients are diffuse large B-cell lymphoma, about 1/3 are follicular lymphoma, and then we have a smattering of other diagnoses, marginal zone lymphoma, mantle cell lymphoma. In follicular lymphoma, we see an overall response rate of 95% with a complete remission rate of about 77%. In diffuse large B-cell lymphoma, we see a dose-response relationship where we've been seeing higher response rates as we go up in dose. And so when we look at patients above a dose of 80 milligrams with this drug, we have looked at the large cell lymphoma patients who've been previously exposed to CAR T therapy and those who have not. In the patients who have not had prior CAR T therapy, we see a 71% overall response rate, and those were all complete remissions. In the patients who had prior CAR T therapy, we see a 50% overall response rate, and about half of those, or 25%, were complete remissions. So very active in these patient populations. And we see about 2/3 response rate, 67% in these additional diagnoses of mantle cell lymphoma and marginal zone lymphoma. And in each of those diagnoses, we also see about half of those patients, or 33%, having a complete remission. So again, in this phase 1 study, we're seeing activity signals with what seems to be a safe drug.